Agriculture, plant cell and cell organelles, chloroplast, mitochondria, ribosomes, Golgi complex. Chloroplast. Chloroplasts are organelles found in plant cells and other eukaryotic organisms that perform photosynthesis because of the presence of green pigment, chlorophyll. They are flattened discs, usually 2 to 10 micrometers in diameter and 1 micrometer thick. The chloroplast is surrounded by double-layered membrane. The space between these two layers is called intermembrane space. Stroma is the aqueous fluid found inside the chloroplast. The stroma contains the machinery required for carbon fixation, circular DNA, 70S ribosomes, called a semi-autonomous organelle, etc. Within the stroma, the stacks of thylakoids are arranged as stacks called grana. A thylakoid has a flattened disc shape and has a lumen or thylakoid space. The light reactions occur on the thylakoid membrane. Functions of chloroplast. 1. The important processes of photosynthesis, i.e., light and dark reactions, occur within the chloroplast. 2. The granum is the site of NADP reduction, forming NADPH plus H+, and photophosphorylation, i.e., formation of ATP in presence of light. Thus, light reaction of photosynthesis takes place in the granum region. 4. The stroma is the main site for the dark reaction of photosynthesis. 5. The chloroplast has its own genetic system and is self-replicating. Mitochondria. Mitochondria are rod-shaped cytoplasmic organelles, which are main sites of cellular respiration. Hence, they are referred to as powerhouse of the cell. Each mitochondrion is enclosed by two concentric unit membranes comprising of an outer membrane and an inner membrane. The space between the two membranes is called peri-mitochondrial space. The inner membrane has a series of infoldings known as cristae. The inner space enclosed by cristae is filled by a relatively dense material known as matrix. The matrix is generally homogeneous, but may rarely show finely filamentous or fibrous structures. The matrix contains several copies of round or circular DNA molecules and 70S ribosomes. It is also called a semi-autonomous organelle. Functions of mitochondria. 1. ATP, the readily available form of energy, is produced in mitochondria. 2. Krebs cycle takes place in the matrix of mitochondria. 3. The enzymes of electron transport chain are found in the inner membrane or cristae of mitochondria. 4. Heme synthesis occurs in mitochondria. 5. Controls the cytoplasmic Ca2+ concentration. Ribosomes. Chemically, ribosomes are ribonucleoprotein (RNP) complexes. This is a membrane-less. Ribosomes are of two types. Ribosomes of prokaryotes have sedimentation coefficient of 70S and consist of two subunits of unequal sizes, 50S and 30S subunits. Ribosomes of eukaryotes have 80S sedimentation coefficient, 40S and 60S. The two or more ribosomes become connected by a single mRNA and then may be called polyribosome. The major function of the smaller subunit of ribosome is to provide proper site for binding of mRNA and its translation. The larger subunit of ribosome supports translation and translocation processes coupled with polypeptide synthesis. Functions of ribosomes. 1. They provide the platform for protein synthesis. Point 2. They have the machinery for protein synthesis. Golgi complex. Golgi bodies is an assemblage of flat-lying cisternae, one above the other in close parallel array. Each Golgi complex has 3 to 12 interconnected cisternae which are composed of lipoproteins. Functions of Golgi complex. 1. It helps in packaging of proteins for exporting them. 2. It plays a role in sorting of proteins for incorporation into organelles. 3. It is involved in the formation of the cell wall of plant cells. Thanks for watching.